Hello. This afternoon, we will be talking about the enzyme nuclease. I am Elisa Meyer from the Biochem 2 class, Biochem 316, and this is our individual project number 2. Here is an example of DNA and RNA and what nuclease does in them. I will come back to this picture quickly, but first I'd like to read a few bullet points so that this picture is better understood. So what is nuclease? Nuclease is an enzyme that cleaves the chains of nucleotides in the nucleic acids into smaller units. For later, we will see that what that statement means is nucleic acids, when it comes to digestion, when it comes to the progression and help with medicine and medical treatment, as well as everyday things in the body such as DNA and RNA splitting. And this nuclease is an enzyme that's capable of cleaving the phosphodiester bonds between nucleotides of nucleic acids. So if we go back to the picture, we see up top, figure number 1 is a full DNA. Picture number 2 is where it's its split, and picture number 3 is where it's been broken down and split into a smaller piece. Nucleases are critical components to biological processes involving nucleic acids. So some nucleic acids are DNA specific and some are RNA specific, so they're assigned to their specific things, and some can degrade both DNA and RNA, so there are several different nucleases within the body. There are 4 kinds of nucleotides. Those nucleotides are what make up the nuclease, and they are found in DNA and RNA. They are called adenine, thymine, cytosine, guanine, with uracil replacing thymine in RNA. Each nucleotide has the ability to match up with each particular nucleotide that it needs. And when those two are able to come together, they form the complete DNA or RNA strand that's needed in the specific moment. For an example, when forming a DNA ladder, adenine will line up across from thymine, and cytosine will line up across from guanine. In RNA, uracil matches up with adenine. So its effects on the digestive system and environmentally, both inside and outside of the body. In living organisms such as humans, animals, bacteria, plants, well, plants are a bit different, but they are essentially machinery for many aspects of the DNA repair. Defects in certain nucleases can cause genetical instability or immunodeficiency. And so this within humans can cause defects when born and can cause a weak immune deficiency, causing many health issues within the newborn child or even at times an adult. Nucleases are also extensively used in molecular cloning. So that is something we'll go into more when we talk about medicine in just a few minutes. But because it has the ability to break and bond within DNA and RNA, they're able to use this enzyme to kind of sit in where bonds are needed and to speed up the process when it comes to cloning. So they're able to create false DNA cloned from the enzyme nucleus. The pancreas is the biggest finding that I found with nuclease. So because our pancreas releases because the pancreas also releases nucleases, those digestive enzymes need broken down. And they get broken down into nucleic acids like DNA and RNA within the nucleotides. So this is where the building blocks of nucleic acids happen. But we need this enzyme to break down those nucleic acids so that they can properly become the DNA and RNA that's needed. When these nucleotides reach the ileum, the last section of the small intestine, they are further digested into sugars, which are bases and phosphates. When those changes happen, there has to be an appropriate pH change. Our body is constantly fighting to heal, produce, and to be in homeostasis. So in order for this enzyme to properly work where it needs to, as we just spoke of one example in the pancreas, the pH has to change and alter to the degree of ionization where the amino acid R group in the protein, as well as the extent to which they can bond with the hydrogen. So to give an example, the pH within the stomach is we'll say the pH of pepsin is 2. If enzymes need to be broken down by this, if enzymes need to break down, that pepsin will have to go to the same pH as the stomach, so they all have to alter going to a pH of a level 2 in order to properly work. There also has to be the proper temperature in the environment for the enzyme to work appropriately, and that is called the appropriate temperature, or the modified temperature. So everywhere in the body, things are constantly changing in order for enzymes to work, in order for their result of their production to work. So nucleus digestion and sedimentation in sucrose gradients can be used to conform the formation of nucleosomes. Without the enzyme nuclease, substances in the duodenum would not be properly broken down, as it aids in digestion for breaking down nucleic acids. And so again, we did mention that already. This is just a bit of a summary, but going back to what I just said, everything has to be in the proper pH, the proper temperature, in order for the enzyme nuclease to properly work. And if they're not, then issues can result. So here, again, it's called the temperature optimum, but we already explained that, so we'll move on to the next point. Something else that the temperature optimum can do is, if it is above temperature and it is increasing temperature, that begins to also increase the vibrational energy of bonds within an enzyme, and eventually that can create or cause bonds to become very weak. Their interactions are disrupted, and the enzyme can become denatured, which would cause any reaction to stop and them not to be able to properly perform their job. So enzyme of nuclease and general enzyme use in medicine. DNA technology has provided medical science with yet another perhaps more promising clot-dissolving enzyme. So it's playing a role in clot enzyme breakdown, which is very important because unfortunately clots can result from all sorts of things. But what first comes to my mind and what I've read as well as what I've been studying is, with how sedentary we are, a lot of blood clots happen. But when we can have an enzyme that can help clot dissolving, it helps within health, which then results with reducing hypertension, heart disease, or cardiovascular disease, which in turn goes into heart issues. Another thing in medicine is elevated blood serum concentrations of the enzymes amylase and lipase are indications of the pancreas. So as I mentioned, I read most about the pancreas for our enzyme that we're specifically speaking of. An inflammation of the pancreas is a huge thing known today, both within the United States and across the world. Pancreatitis, cirrhosis, and hepatitis are the levels of those diagnoses are huge. So us finding an enzyme that can help with liver disease and help with reduce cirrhosis and hepatitis is a really big finding. And it does so by, when long DNA fragments are used for reconstruction, they are reconstituted material that can be digested with micronucleus to show the nucleus-sized DNA fragments and protect from digestion and breakdown and enzyme prohibition. Enzymes are also increased in concentration following a heart attack. So as I spoke, it all starts with something as simple as a blood clot or hypertension, but heart disease, cardiovascular disease, is one of the #1 killers within the United States and the world right now. So physicians have been able to find an enzyme that can help reduce heart attack, and they've had to do so through many studies, but something that they've had to really study and do is being able to tell the difference or differentiate between the two conditions by considering relative increase in two separate enzymes. Enzymes are also used as analytical reagents in the clinical laboratory, owing to their specificity. Enzyme replacement therapy can also be used in the treatment of certain diseases. And here are my sources. I hope you have a great day. Thank you.